So in speaking with Meg and other members of the Alliance, something that I've noticed also outside of AE, but with respect to patients in general, is that nobody ever has like the opportunity to explain to patients or their families what's actually happening to their body on like a biological level with their disease. How can that biology affect, the symptoms that you see, maybe why one person with NMDA receptor encephalitis has some symptoms and another one has a different set of symptoms, why your symptoms change throughout your disease course, all of these things that are related to biology, but you guys never really get the opportunity to hear about or have discussions with, because a lot of it is all based on clinical stuff. For me, going through AE as someone who understood all of this, I felt like it made certain parts of my journey less frustrating in certain regards, more frustrating in others. But I felt like, this is something that changes your life or your loved one's life pretty drastically. And if there's an opportunity for you or your loved ones to learn something that maybe helps make things less frustrating or positively impact your journey, we should do that. That being said, it is really, really, really hard to communicate science and this sort of stuff to people who do not do this every day. And that is no fault of your own. It is actually our fault. We need to do better at that. As part of this presentation, I have tried really, really hard to make sure that everything we talk about on a biological perspective is in some way relatable to things that you maybe interact with or know about or are familiar with on an everyday basis. That being said, this is what I do every day. I've done it for almost a decade now. And so things that maybe sound like they're relatable might not be, because the conversations I probably have at lunch or in the hallway are using words that maybe you've never heard of. So I want this to be all about you guys. The whole purpose is that it is a positive experience for you. I have two parts of the presentation. The first part is about kind of the brain and the nervous system. The second part is the immune system. If we get to the end of the first section and you're like, this is not helpful and not good information and not a positive experience, we do not have to go on. If it's been too much, we also don't have to go on. But this is all about you guys. So I'm pretty flexible and we'll just get started. And as Meg said, please put your questions in the chat box. And if there's anything there are no stupid questions. If I'm not doing a good job explaining something or if it's confusing, like please point that out, because I have no problem trying to do a better job at that. So I will, let's see, share my. Okay, can everyone see this?
Yes.
Okay. So as Meg said, I am a postdoctoral research fellow at the Indiana University School of Medicine. I study a rare cancer predisposition syndrome called neurofibromatosis type 1 and type 2. Most of my work over the last decade has involved the nervous system and the immune system and the intersection of the two. And so, as I said, that the goal of this presentation is to provide, a better understanding of the biology for you guys and how it relates to the AE experience. The caveat of this presentation is that these are all really, really complex topics. And while the information presented here is based on the best currently available, data on the topics, it has been simplified. So it's not necessarily complete or in its entirety. The other caveat that relates to that is there is a lot that we do know, but there's also a lot we don't know. And that's within the field of medicine and science in general. And there's also a lot that I'm still learning. So even if we do know something, I can't possibly know everything about the immune system or the nervous system. And so, as we're sitting here right now, somebody is discovering something that will change part of this presentation. And the last caveat is that, medicine and science are rarely black and white. So everybody's body is different and everybody's situation is different. And so we can't really treat anything as concrete, black and white, ideas, but they are in fact all, true and factual and accurate. Oh, it's not letting me. Oh, there we go. So this is kind of the outline of the presentation. I won't go through all of it. Basically, part one is going to be about the nervous system. Part two is about the immune system and how both of these things relate to AE. And again, reminder, this is all about you guys. So I want it to be a positive experience. And if there's anything that I can do either after the first part or in the future, not limited to never do this again, that's totally fine. Be honest. Again, this isn't about me. It's all about you. So for our first part, we'll talk about, neurobiology basics, which is basically just, about the brain and how it works under normal circumstances, and then how that relates to AE. So most of us have seen a brain, if not our own or our loved ones. But what we probably haven't seen are the parts of the brain that you can't see on imaging. And that's the cells that make up the brain. So this is just kind of a cartoon. If you were to look at the brain under a microscope, you would see these different cells. The gray cells are called neurons. And then there are cells in between them called microglia and astrocytes. And collectively, they're called glial cells. So we can actually see these under a microscope. And I haven't taken this picture, but this is a lot of work that I do is with microscopes. But we can actually see in real time how these cells interact with each other and the characteristics of these cells. And there are multiple different ways that we can see that. And we can see different degrees of detail, more or less. We can sometimes see these in real time, like while the cells are still alive in a dish and how they are moving. So this is how we know a lot of the information, is that we can actually see these things and we can change these things with scientific techniques. So the neuron is the functional unit of the brain and the nervous system. And the way that neurons carry out their functions are through signaling. This is basically just communication. And the way that neurons communicate with each other is different depending on the type of response you want. Do you want to have a thought? Do you want to have a feeling? Are you trying to maintain a heart rate? Or are you going to, go off for a run? The way that neurons are able to communicate with each other are through receptors, and some of you may be recognizing some names on here. So receptors are basically like mailboxes, and they are on the surface of the cell. So the first neuron wants to tell something to the second neuron, and they will send a message, just like you or I would send a message. And they can send many different types of messages, much like you and I can send, an email, a text message, phone call, face-to-face communication, shouting. Neurons can do the same thing. They have many different ways to communicate with each other, either close by or far away. The neuron that they're trying to get a response from, so neuron number 2, has what's basically like a mailbox expressed on the surface. And so they will receive the message, and that message then has instructions on how they're supposed to respond. And it ends up being this sort of domino-like cascade. But these receptors or mailboxes, for many of you are familiar. And when you have something that disrupts this, then you have a disruption in the messages getting, through to the second neuron. So another way to think about this is through a lock and key type mechanism. So we call it a ligand. That's just a fancy word for the message. Can be thought of as the key, whereas the receptor, so something like NMDA receptor, is basically a mailbox or a lock. What this looks like is that the key and the lock are separated, but then they come into contact, just like you would put a key in your car ignition and turn it on. This is the same thing. The ligand makes contact with the receptor, and it causes the neuron to turn on. This is a little more complicated than just on and off, because it's not necessarily just neurons are on or neurons are off. We actually have broadly 2 different types of neurons. We have ones that are excitatory, which means that when you turn it on, they start the car. And then we have neurons that are inhibitory. And when you turn those on, they stop the car. This can kind of be thought of as like a gas pedal in your brakes. And the ability to have different responses is related to the balance of these 2. If you just slam on the gas and you never use your brake, you're not going to be very happy with the end result. And if you only keep your foot on the brake, you're also probably not going to be happy with your end result, because you'll never get from point A to point B. The same is true with neurons. It's a balance. So their communication between each other is balanced by these stop and go signals. Most of you are probably a lot more familiar with this process than you even realize, because a lot of the medications that we use to treat AE and other neurocognitive neuropsychiatric disorders actually influence this process. So seizure medications, for example, a big class of seizure medications that you've probably heard of, at least one of these, influences the first neuron and stops it from sending a message to the second one. So this is kind of like a break. These would be like your breaks. You're saying, you know what, there's too much activity, we're going too fast, we need to slow down. That's a way that you can kind of think of this. Now, that's obviously very simplified for how these drugs work, but that gives you kind of an idea of how they interact with this process. Another class of drugs are mood stabilizers or antipsychotics, sedatives. Many of you have maybe heard of these drugs as well. They interact with the second neuron. So they basically shut the mailbox or open the mailbox, depending on what you need. So this is another way that we can influence this system. If too many mailboxes are open or not enough mailboxes are open, then we can give drugs that influence this part of the process. Likewise, antidepressants or anti-anxiety meds like SSRIs or SNRIs, they also can influence this process by increasing or decreasing the number of emails you get in your inbox or in your number of postcards you get in your mailbox. So this whole schema of basically how neurons talk to each other is not only super important for how you get to live your life and the actions, thoughts, emotions you get to experience, but it's also really, really important when there are disruptions in those, because we can actually do something about it. So neurons are really sensitive. They are really, really high maintenance, and they very much need a high maintenance or a optimal environment. These are not the type of cells that are like going to go camping with a tent and a backpack. They are going to stay at the Hilton, and they want room service. So but part of that is because they're so highly specialized and that they're responsible for so many important functions that they have to be so sensitive. This is what allows them to perform these very, very precise and really, really important functions. The fact that you're able to have a single type of cell control both your heart rate, your thoughts, your talking, your walking, to be able to do that is really, really complex. And so their environment has to be maintained at a really optimal state. And the microglia that we talked about earlier, these are kind of the supportive cells. And so they work really, really hard to maintain this happy and safe environment that maintains this optimal neuron performance. And you can kind of think of the neurons as like a professional athlete or an Olympic athlete, whereas the microglia are the coaches, the trainers, the nutritionists, administrative assistants. They're the ones that kind of make sure that the neuron is living the cush lifestyle that it has become accustomed to. So any changes in this environment, like infection or inflammation, can actually influence the neuron's function directly, either by acting directly on the neuron, or it can cause a change to the neuron's function indirectly. And what do I mean by that? So what I mean is that the support cells, so the microglia, if they sense a change in the environment, they're the first ones to respond. But that response can actually compromise, neuron performance. And this is kind of like a Goldilocks phenomenon. You need them to respond because you need them to protect the optimal environment of the neuron and the neuron itself to avoid getting, attacked by pathogens, viruses, bacteria, toxins, poisons. But you also don't want them to respond so harshly that they cause damage to the neuron. And under normal circumstances, your body is under threat, goes through many changes a day. This process goes on without a hitch. Conversely, sometimes it doesn't. And you end up getting what can happen is neuron dysfunction or injury. And it doesn't have to, it's not black and white where if one thing happens, like say you get a virus, you only get neuron dysfunction, or if you only get hit with the toxin, you only get injury. It can be on a spectrum of dysfunction to injury. And the effects of the dysfunction versus the injury, whether it's permanent or short-lived, that can also change. But these are two effects that can come out of an appropriate or an inappropriate response. So when we talk about dysfunction, what does that mean? Well, this takes us back to our receptors that our mailboxes and our message sending between our neurons. So when we talk about dysfunction, we're talking about any part of this process that is disrupted. So this could be the first neuron is unable to send the message at all, the message is unable to enter the mailbox, or the mailbox just simply does not exist. So how does this relate to AE specifically? And there are several subtypes of AE that we actually know are caused by this type of dysfunction. And GABA B receptor encephalitis is one of those. So in this setting, the antibody binds the receptor or the mailbox, and it basically prevents the message from entering. So this is like you would prevent the key from entering a lock. The key is still there, the lock is still there, but they cannot interact with each other. The same is true, your mailbox is frozen shut. Another way that this can happen is that you decrease or completely eliminate the mailbox. So basically someone has just plowed over your mailbox, it no longer exists, it's in smithereens on the ground. This can happen with LGI1 encephalitis. And so what happens here is the antibody binds the LGI1 protein, and it prevents the communication between the two cells. This would kind of be like if you had an old email account and you just stopped checking the emails because you weren't getting sent any. So you decide never to like open the email address or email inbox anymore. So the receptor then goes away, just like you ignore that the email goes away, the email inbox goes away, or you don't live at your house anymore, so you've stopped checking the mailbox. Another version of this is with NMDA receptor encephalitis. In this case, the antibody binds to the receptor, to the NMDA receptor, and it actually causes the neuron to engulf or eat the receptor. And so it takes it away from the outside. And that decreases the number of mailboxes available to receive messages. So in both of these scenarios, you have a problem where either your mailbox is there, but it's not functioning the way that it should, or your lock is there and it's not functioning the way that it should, or you no longer have any mailboxes or locks. So another way that neurons can experience responses or be affected by responses to change in their environment are through injury. And most of the time what happens is there is a intracellular, that means within the cell, antigen. And an antigen is anything, doesn't matter what it is, anything in the entire world that causes an immune response. So what happens is that antigen is presented or like shown like a flag of a foreign invader on the cell surface, and the immune cell recognizes it and launches an attack. This is the process that happens if you were to get COVID-19 or a virus of some sort or a bacterial infection. This is the same process. The only difference is it's not supposed to be doing it to your own cells. Another thing that can happen is that the antibody recognizes the flag on the cell and it activates this system. It's called the complement system. You don't need to know any details of that. The only thing that's important here is that this has been recorded in the literature to occur specifically, right now, it's one of the only ones we know, I believe, with Caspr2 mediated encephalitis. So the point of even bringing that up is just to show that with different types of antibodies, you can have different types of responses from the immune system. It's not just the same thing. And there are reasons why that happens. And that can be important for when we decide what medicines are going to be given to patients, right? Because if all you have is a problem with the complement system, then it's not really going to do you a lot of good if we, take out a different part of a system, like this immune cell attack. Maybe that's not your problem, or maybe your problem isn't the antibodies as much as it is, T cells. So this is something that is really important for how we determine which patients get which medicines. After this process happens, the neuron is injured. And that can be permanent, or there can be reparative mechanisms that come into play. So one of the things that's really important is that we don't necessarily know exactly all of the details of which antibodies cause which side of the spectrum, but we do kind of largely know that. So we do know that certain antibodies are more likely to cause dysfunction, and I've listed those here, whereas other antibodies are more likely to cause injury. And then GAD65 and several others kind of fall in the middle. I've read a lot about the literature about GAD65 recently, and it seems like the consensus is that we're not as sure about what's going on there as we maybe once thought. So, but again, this can be a spectrum. So just because some antibodies are more likely to cause dysfunction, that doesn't mean that they don't also have components of injury, and vice versa. So how does this relate to AE symptoms? So the brain is organized in functional compartments, and different parts of the brain have different functions. So, and it goes from like large to small, right? So you have all of these large lobes, and they all have different functions that are specific, and we actually know what those functions are. Even within those lobes, you have areas that have even more specialized functions. And again, we know what those functions are for the most part. Further than that, you have even smaller areas that have specialized functions. So there's many different layers to the functions that your brain can perform. And how is that possible? So the first thing we'll go over are what are the major functions, because many of you have either had loved ones or yourself have probably experienced changes or symptoms that are related to a lot of the functions of these lobes. So the frontal lobe is mostly involved in planning, problem solving, your personality, movement, recognizing and regulating emotion. The parietal lobe involved in sense of taste, touch, smell, math, reading. Many of you may have had trouble with math or subtraction. That would be because you have involvement of your parietal lobe. If you are having trouble discerning your right from your left, or if you think that if you don't even really recognize that you have two sides of your body, that can be due to damage in the parietal lobe. Your occipital lobe is mostly involved in vision. So there are case reports of patients with anti-NMDA receptor encephalitis who have involvement of their occipital lobe, and they will have changes to their vision. And then the one that you're probably most familiar with is the temporal lobe, where you would have trouble with language, memory, behavior, mood, emotion. This is also where you process sights and sounds. So you may be hypersensitive to sound. You may be hypersensitive to sight. That's because the process that is disrupted are in these very specific areas. And so when you say to me or someone who knows the way that the brain works, you say, I'm having trouble with memory or processing sound. That gives us kind of an idea of where you're having trouble. Beyond that, you've probably heard of the limbic system. And the limbic system is just an even deeper layer of functional division, I guess you could say. And it's composed of different parts, including the hypothalamus, which is involved in regulating thirst, hunger, hormones, your heart rate, your blood pressure. So some of you may have had differences in your heart rate. You may have had heart palpitations. You may have had fluctuations in blood pressure. You may have had children or yourselves experience difference in your menstrual cycle or growth. And that's because you have involvement of this area. The amygdala is another portion. It's like a bean. It looks like a bean. And this is another area that you can have difficulty with and processing fear and danger. This is where your response comes in. Regulating emotional behavior. So, maybe responding inappropriately to something. And then your motivation. Maybe you don't have any motivation to get up and get out of bed or eat something, take a shower. Damages to your amygdala can cause all of these different types of symptoms. Your hippocampus is another portion that is involved in the limbic system, and it's mostly involved in emotion and learning and memory. So if you're having trouble with learning or memory, you may have involvement of this area. And then your cingulate cortex kind of integrates all of these things and puts them all together into actions or thoughts or feelings, and then stores them away. So how is this possible? How is it possible for different parts of the brain to have different functions? Well, just like a business has different layers, the same is true with the brain. So if you think of your brain as the company and each area of the brain as a specific department, within each department, you have all these different employees. And while, let's say they're part of the marketing department, their goal is to do all things marketing, right? But even within that marketing department, you're going to have individual employees who have expertise or unique abilities. And that's the same thing with the brain. You can have different types of neurons in different locations that allow these different areas of the brain to perform these different functions. So again, the reason for these different functions are neuronal diversity. So you have a diverse group of neurons that make up these very specific parts. And the reason that one part of the brain is different than another is because of the specific mixture of neurons that you have at that location. Another way that you can have diversity is, again, through this receptor program. So receptor diversity, all that means is that some neurons maybe have 25 NMDA receptors or mailboxes on their surface, whereas other types of neurons don't have any. So this is what allows you to have this diversity and have different responses. For the purposes of AE, this is what's really important. So most of you have probably seen one of these before. This is not a mistake, it is a weather map. But if you haven't seen one of these before, the red area or the darker orange, yellow, basically that means you have more rain or a bigger storm, right? There's more happening there. Whereas the green or the blue means there's less happening, or the storm is less severe, there's less rain. So what's really, really cool is that we can actually do this with the brain. And we can see the number of, or the receptors on your brain and where they're located by doing a technique that is similar to this. So the next thing I'm going to show you, I don't want it to be overwhelming. It's the same concept, it's just the brain instead of the state of Indiana. So this is, these are sections of the brain, and all it's showing you here are the different receptors or mailboxes like we've talked about. And what you can see, the whole point of this is that their NMDA receptors are located in overlapping areas with others, but they also are located in different areas. If you look at the dopamine receptor, there are a lot of red in one area and then not a whole lot in other places of the brain. So this can kind of give us a clue if you have a specific type of antibody, or if you have specific type of symptoms, but we don't necessarily know what antibody you have. This can kind of give us a clue as to what's going on, because we know where these receptors are largely located or where they're most commonly located. So this is kind of like the game of Clue, where your suspects are your antibodies, and then the rooms are the different parts of the brain, and then you have your symptoms. So it's anti-NMDA receptor antibody in the amygdala, and you're having psychosis, or GAD65 in the temporal lobe, and you're having seizures. So this is, in fact, a lot of times what I do when people are telling me their story. I basically have a game of Clue in my head. So what this would look like with respect to AE, if you have impaired NMDA receptor signaling, if your NMDA receptor or mailbox is not functioning, and it's not functioning in the amygdala or the hippocampus, you might have impaired memory, you might have impaired regulation of your emotion, you might have abnormal fear responses, you might have changes in motivation. Another way that we could look at that is if you have impaired NMDA receptor signaling in your hypothalamus, You might have changes in your heart rate or heart palpitations. You might have abnormal thirst or hunger. You might have changes in your endocrine system which would present like growth disruption or changes in a menstrual cycle. These are all things that can happen, and there's a biological reason for that that we've just kind of gone over. So what about the fluctuating symptoms, right? Because you think if somebody just like plowed over your mailbox, that's going to be a thing that's kind of permanent, right? Fortunately, the receptors in the brain are a little bit more sophisticated than your average mailbox. So ways that we can kind of theorize or possibilities as to why you would have differences in symptoms, even from day to day, patient to patient, hour to hour. One mechanism is that cells can actually make new receptors to replace the ones that have been removed or injured by the antibody-mediated process. That being said, if someone is just going through your neighborhood and just running over every mailbox in your neighborhood, you can definitely replace them, but they can probably run them over faster than you can put up new ones. So you wouldn't necessarily get all the way back to normal, right? You would still have some symptoms, they just might be less severe or they might be different. Another reason you could have fluctuating symptoms or differences between patients is that physical, mental, and emotional stressors. So these all produce biological responses in the body. It's not just magical gnomes or unicorns. That it's an actual biological response that is produced when you are subjected to mental, physical, emotional stress. When your neurons are healthy, they're like, we got it, not a problem. We can handle a little bit of a challenge. When your neurons are sick, this is just like when we're sick, right? It's really hard. Well, I find it hard to run a marathon even when I'm healthy, but it's really, really hard when you have the flu. So this is kind of the same thing. They kind of got it, like, depending on how sick your neurons are, we got this, but it may not be pretty. So if you are overstimulated by a stressor, you might be able to handle it, but it might not be your normal ability to handle it. Conversely, if you're super sick, we do not got this. So it may be harder to handle, longer periods of stress, more intense stress, whatever it may be. Another reason that you could have fluctuating symptoms would be compensation. So your body, its primary goal is to maintain optimal performance at all times. The same is true with your brain. And so the body is not designed to fail easily. And a way that it can avoid that is by having compensation. So other employees within the corporation can pick up the slack. And also, but you can also have the same neuron depending on how sick it is. So, right, when you or I are sick, we can compensate in certain ways, but sometimes that compensation goes too far. And this would be like when you gun it in your car or when you slam on the brakes. So the response you get is maybe an exaggerated response to the stressor, or, I don't know if anyone has a loved one or themselves has experienced, heightened abnormal anxiety or fear, or, something that should elicit anxiety actually elicits, fear or laughing. These are things that can happen when your brain is sick and you're trying to compensate for that, especially when it's subjected to another layer. So this is the end of the first portion about the neurobiology. We got to start by just knowing what is the purpose of the immune system, what even is the immune system. So the immune system's job, the two big jobs, are to protect against invaders and then to maintain your environment to support peak performance of the body. Just like we just talked about the brain and the glial cells, the body also likes a optimal environment for peak performance. And the immune system is what does that purpose for the rest of your body. It also does it for the brain, but for the purposes of this, we'll kind of separate them. So again, we had already previously talked about this. It's the same concept that the neurons are supported by microglia, and these are the cells that maintain this optimal environment. The same is true of the immune system, which includes the organs of the immune system, the cells of the immune system, which are largely white blood cells. Many of you have heard B, T cells, plasma cells. And then also proteins and chemicals. And for the purposes of our conversation, we'll talk about antibodies and things that are called cytokines. Now, you may actually, so cytokine is kind of an abstract term, but many of you may have heard them with respect to COVID. Another thing is that you already know what these are because you've already learned what the messages are for the nervous system. And cytokines are basically the same thing for the immune system. So if you know what a neurotransmitter is, dopamine, serotonin, adrenaline, that's kind of like a cytokine in the nervous system. And these are different for the immune system. So white blood cells, there are many. Two that are probably most familiar to you are T cells and B cells. These are just two different types of white blood cells in the immune system. B cells can actually grow up and become plasma cells. And plasma cells are the cells that secrete antibodies into your spinal fluid or into your blood. So many of us have probably had a lumbar puncture and we've had antibodies detected, and they're coming from these guys. But before they came from these guys, they started out as a B cell. There are many other types of immune cells and white blood cells. We don't have to go through all of them, but the point is that there are lots of different cells. And that all of these immune cells have a specific area of expertise. So you can kind of think of it as a neurologist versus an orthopedic surgeon. They're both doctors, but they both have different areas of expertise. And some immune cells can perform the same tasks, even though they do have a specific area of expertise. So just like both a neurologist and an orthopedic surgeon can prescribe medication or take blood pressure, the same is true of immune cells. So for a clinical correlation, something that you guys are probably familiar with, you've probably all, you or your loved one or both, have had a test done for your blood or your CSF called a complete blood count. This basically looks at the different cells in your blood, your white blood cells being a subset of those appear in WBC. Beyond that, you can have what's called a differential. And this is basically telling us the exact type of cells that make up the blood. So we can actually see these guys. You can see they have different shapes. They may have different colors. We can actually see these, and here's a picture of what that would look like. And we can tell you what type of cells are what percentage. And this can kind of give us clues as to maybe what's going on in the body, because these cells all have different functions. So some of these cells are known to respond to like allergies. So if you have a really high level of those cells, you may be having an allergic reaction. Other types of these cells, for example, an eosinophil, that's a cell that would respond to like a parasite. So if you have a lot of eosinophils in your blood, you might have a problem with a parasite. This is actually my test. And as you can see up here, there's a flag that says hi under neutrophils. And this actually tells us that I am on steroids. It can tell you that you have a bacterial infection, but we know that the reason that this is high was because I was on steroids at the time, and that can increase the neutrophils in the blood. So this can tell us a lot of information, and we can see all of these things. So how do these immune cells then perform their functions? And one way that they can perform their function is to interact directly with each other. And they can interact with a bunch of different immune cells. They have these little appendages, and they can come in direct contact. Another way is that they can interact with non-immune cells, like neurons, and they can launch an immune attack. This can also happen when you have a viral infection. They will attack the virally infected cell. So this will depend on what your body needs at any specific time and place. Another way that these cells can or that the immune system can carry out its function is through sending messages, just like we talked about with neurons. And they do this by cytokines, which are these small proteins that are released by immune cells. And they can send messages to the other cells that tell the other cells how to respond to any particular event. So they can influence the behavior of cells that are nearby, which you see here with A. They can send a message to cells that are far away through the blood. And then they can also send a message back to the cell that produced them. And they do this again by this lock and key like mechanism where they enter their mailbox. So it's the same exact thing that we talked about with the neurons. These cytokines, though, can have opposing, completely opposite functions. So they can promote inflammation, and they can also prevent inflammation. They can behave like an anti-inflammatory, like the body's Advil. So a single cell can produce many different types of cytokines, just like you can produce a phone call, email, postcard, text message. The same is true with these types of messages. And then different types of cells can then produce the same cytokine, just like I can send a text message and Meg can send a text message. So cytokines are basically just ways that the cells can communicate with each other to tell them how to respond to any given event. And again, they do this by this lock and key mechanism that we've talked about before. Now, some of you may be familiar with the drug tocilizumab or Actemra, and it influences this process by stopping the interaction between between the key and the lock. So it stops the cytokine from being able to enter the lock. And that's how Actemra works. Another way that the immune system can exert its function is through antibody production. And so you can kind of think of T cells as like a TSA dog at the airport. And then you have these cells called antigen presenting cells. And again, we talked about how an antigen is just any substance that triggers an immune response and antibody production. And this is kind of like the COVID antigen test. Many of you might have heard of that. And basically what happens is the antigen presenting cell is like the trainer for the TSA dog. And it takes the antigen and it shows it to the TSA dog and it says, this is what we're looking for. The T cell then goes into the airport, having known this is what I'm supposed to be looking for. And it goes in with its handler and it says, I have found this, and it alerts that it's found it. And the handler is the B cell. And the B cell is like, okay, my TSA dog has alerted, there's a problem here. And so they respond by making antibodies to address the problem. This would be like the handler going in and taking you away to a room and opening your suitcase and going through it. And they are trying to intervene and stop the problem. It's very similar to what you see with the TSA dogs in the airport. Those antibodies, though, then enter circulation, which is not exactly similar to the TSA dogs in the airport, but these antibodies enter circulation, and that allows you to have an immune response all over your body, not just within the point of entry. So how does this relate to AE? So responses to change in the brain, and what that response is can be different depending on what maybe triggered autoimmune encephalitis. Was it an infection? Was it a genetic problem where you created an antibody, a neuron? Was it a tumor? Many different triggers, and we don't necessarily know exactly what triggers AE in everybody. But the basic trigger is that there's a change in the environment within the brain, whether that's an infection or an autoimmune disease that has a non-infectious etiology. It's just a change in the environment. The cells in the brain respond, mostly microglia. They're the first responders. So they respond, and they respond by recruiting and activating immune cells from around the body. So this is basically like they've mobilized the troop. The microglia, the brain, we'll call that Washington, and they have now mobilized the troops, and the Army, the Navy, they're ready to go. So that's the immune cells. They've been recruited, they're activated. These activated and recruited immune cells, as well as Washington, release a bunch of substances like cytokines that will further help with the response. So these cytokines may recruit more immune cells. They may activate more immune cells. They may directly kill the invader. But this is just another part of the response. And then antibody production. Antibody production can again affect the other parts of this process by stimulating cells to release cytokines. They can stimulate other immune cells to come and directly kill an invader. So this whole process occurs. And then you get, in the process, you might get some dysfunction or injury to your neuron. Unfortunately, though, as this process continues, it just keeps going in this cycle. The dysfunction and the injury to the neuron is a change in the environment that is sensed by the first responders. They go through their process of eliciting this response and mobilizing the troops, and it just keeps going. The good news is that we can intervene in a lot of these areas and in a lot of these processes to either stop or make them less severe. So one way is that we can calm down the microglia. In the event that the microglia have responded way too much and that the problem is not still present, we can give drugs that can modulate their response and make them kind of go back to their normal, happy, healthy state. And some examples of that are steroids, CellCept, and IVIG. Another drug set that we can give will affect this immune system, both within the brain and within the rest of the body. And so drugs that can serve that purpose include steroids, rituximab, Cytoxan, CellCept, Immurin, tocilizumab, IVIG, plasma exchange. Now, all of these have different ways that they work and different parts of this process that they work on. So for example, rituximab explicitly affects B cells directly. But the effect on B cells can have a secondary effect on other types of cells. Tocilizumab or Actemra, we talked about having an effect on the cytokines. And then plasma exchange would obviously be something that removes the antibodies. Beyond that, we can then give drugs that affect the neurons and try to decrease the dysfunction or injury. So there's probably many of you or your loved ones have been on a huge combination of these sorts of things and maybe different ones at different times, but this is how this all relates back to AE. Specifically, we can get into how these drugs work. So this is just an example of specifics in relation to the immune system. And so what you can see here is that the different drugs affect, like, so rituximab obviously affects B cells predominantly, but what you can see is that the B cell and the T cell, because they're interacting, this could also have an indirect effect on T cells. Conversely, Cytoxan, mycophenolate, Immurin, those drugs can affect T cells and B cells, and they may affect them in different ways than Rituxan. And then we have drugs like bortezomib, Darzalex, and then CellCept, which can directly affect the plasma cells. In addition, they can also affect other cells. So it's a little bit more complicated than just, straightforward take out a B cell. But the drugs all have different mechanisms and affect the process in a different way. So we'd already talked about Actemra, this being something that stops the cytokines from exerting their effect. So this is more of an indirect effect on the immune system by switching it from a pro-inflammatory or a lot of inflammatory to globally trying to decrease that and make it more of like an anti-inflammatory environment. There may be some other drugs that you've heard of. There are specific drugs that hit very specific cytokines. And then steroids can stop the release of cytokines from immune cells as well. So steroids actually kind of hit all the things, but they hit them in kind of a global way. So that's probably why a lot of you have seen that you get steroids and rituximab, steroids and IVIG, steroids and something else. So this is how we can give all of those drugs in combination because they all have different strengths and functions, but they're all important for kind of combating the problems with AE. So I won't go into much more detail with the immune system. I figured this was going to be our first kind of, if it went well, our first session. But this kind of gives you an idea of things that we could, if you want to have future sessions, talk about. But to summarize kind of what we have talked about today, are that many factors can contribute to differences between patients, including what the triggering event is, what type of antibody, the antibody titer, the type of response. So do you have dysfunction versus injury, the severity of your disease? And that can be reflective of, how many antibodies you have or like the severity of the response. It can also be reflective of, do you have more injury or more dysfunction, or do you have somewhere in the middle? The severity can also be affected by the duration of the symptoms. So, did you have dysfunction at the beginning, and now it's gone on for quite some time, and now you have a little bit more injury? you can also have differences because of, the treatments that you maybe get initially, and then your individual response to treatment. Even if we've picked the perfect drug, bodies unfortunately or fortunately have a way of surprising us. So we don't always, everybody's body is different. And we can't always predict how people are going to respond to the drugs. And beyond what we've talked about today, there are many, many, many more factors that can contribute to any differences that you may see between patients or even within yourself. And so that I hope this presentation has given you a little bit of an insight into, why this is so complicated and all the conversations that we've probably had with each other about these differences. And that is what I have for us today. So we can take questions on, if we want to go more into the immune system, great. If not,
I think what I'm going to do is go ahead and thank you officially on the recording. So whoever listens to this as a recording will know how much we all appreciated it. And again, I look forward to you seeing some of these fabulous comments because everyone's appreciating it. So thank you officially. And I'm going to stop recording and we're going to move on from there.